Usually when we talk about a medical diagnosis, in the modern world, there's this underlying expectation of absolute precision.
Right, like structural engineering.
Exactly. It's like you fall off a ladder, you break your arm, and the X-ray produces that high contrast image, with the jagged white line right across the bone.
Yeah, it's entirely binary. The fracture is either present or it's absent.
And the doctor just points at the illuminated film and says, there is this structural failure.
Right.
We've built this entire medical paradigm around that level of visible, undeniable certainty. We want our biology perfectly categorized.
We really do. But the moment you step out of the orthopedic ward and into the realm of neurodevelopment,
It all falls apart.
it shatters completely.
Yeah.
When you get into cognitive function, attention spans, focus, we are suddenly navigating a diagnostic landscape that is incredibly murky.
Yeah, and we're dealing with symptoms that feel like they plague almost everyone trying to survive our current reality, right? Because we exist in a world moving at an absolutely blistering, unsustainable speed. And the fallout of trying to process all that data often gets swept under this single, massive umbrella diagnosis. ADHD. Attention Deficit Hyperactivity Disorder.
Which is, frankly, the ultimate example of diagnostic muddy waters. For decades, the prevailing approach has been to treat these core symptoms.
The inability to focus, the impulsivity.
Right, that sensation of having a motor running inside your mind that you just cannot shut off. They treat it as behavioral quirks, or even more destructively, they're framed as personal moral failures.
Yes. It is treated entirely as a discipline issue. The societal narrative is just so dismissive. People are told to just try harder.
Just put your smartphone in another room.
Buy a better planner. Focus, But today, we are taking a massive, deliberate detour from that behavioral narrative. And welcome to the Deep Dive, by the way. I'm thrilled you're joining us, because our core mission today is to completely shortcut you to being incredibly well-informed on a framework that is quietly but radically revolutionizing how we view cognitive health.
We're pulling directly from some fascinating analysis laid out in Gary Brecka's Ultimate Human Framework.
Which is just wild stuff. And as always, we're not taking sides or endorsing everything. We're just unpacking the specific, dense biological science he presents regarding ADHD.
Exactly. We're going to look at one specific genetic lever and how highly targeted nutritional chemistry interacts with it.
Yeah, how we might actually be able to feed our brain the exact molecular raw materials it requires to function.
And what makes this specific analysis so vital is that we're actively discarding the notion that these cognitive struggles are just an inevitable consequence of modern environments.
Right. We aren't just blaming TikTok anymore.
No, we're fundamentally reframing these attention challenges. We're looking at them as profound, microscopic nutrient deficiencies driven directly by an individual's unique DNA blueprint.
Okay, let's unpack this, because the scale of this paradigm shift is massive. If ADHD is viewed strictly as a psychological hurdle, the interventions are either behavioral therapy or, heavy pharmaceutical stimulants.
Right, the standard protocol.
This framework repaints the entire picture. It's no longer a behavioral puzzle. It is a deeply biological, genetic, and nutritional equation.
And the implications of that shift alter everything about how we approach human cognition. We're no longer talking about top-down behavioral interventions. We're talking about the microscopic genetic engine that actually builds and regulates the brain from the bottom up.
It's incredible.
But before we can even begin to understand how to optimize our diets for focus, we have to completely demystify the mechanics of that engine.
And that structural reality brings us to the absolute center of today's deep dive, the specific gene that sits at the foundation of this entire biological architecture, the MTHFR gene.
And yes, as the source humorously points out right at the beginning, it is not a swear word.
Even though your brain instantly reads it as one.
It certainly invites a double take, but MTHFR actually stands for methylene tetrahydrofolate reductase.
Which is a mouthful.
It's a dense piece of biochemical nomenclature for sure, but it represents one of the most critical biological puzzle pieces in the human body.
So let's slow down and dissect exactly what methylene tetrahydrofolate reductase actually does, because the text explains that this gene is responsible for a highly specific biological conversion. It produces an enzyme, right?
Correct.
And that enzyme is tasked with taking an amino acid and converting folic acid, along with its derivatives, into a usable active methylated nutrient called methylfolate. I don't want to just gloss over the word conversion here. The body is taking a raw material, and it has to physically alter it to use it.
The concept of biological conversion is paramount. The human body rarely utilizes raw materials in the exact state they're ingested.
Give me an example.
Well, think of the MTHFR gene as an incredibly sophisticated microscopic crude oil refinery. When crude oil is pulled from the earth, it contains massive potential energy, right? But it's structurally useless to your car.
You can't just pour it into the gas tank.
Exactly. You cannot pour unrefined crude directly into a combustion engine and expect the vehicle to move. It has to go through a catalytic cracker in a refinery. It must be subjected to heat and chemical alteration until it's refined into high-octane gasoline.
And our cellular biology operates on the exact same principle.
The exact same principle. The MTHFR gene provides the instructions to build the refinery, which is the enzyme. That enzyme takes the crude raw material of folate or folic acid, and it refines it into methylfolate.
Okay, I see.
Methylfolate is the actual combustible, high-octane fuel that the brain's engine requires to run.
So if we follow that analogy, what happens when that biological refinery is running at peak capacity? We want to understand the actual scale of impact we're talking about here. What does this refined methylfolate actually build within us?
The scope of its influence is honestly staggering. The analysis details how this specific gene, through the production of active methylfolate, regulates the absolute foundational pillars of the human experience.
Like what?
It is the chemical basis for personality development. It regulates your mood and your emotional baseline. It dictates your mental clarity, your cognitive processing speed, and even the fidelity of your short-term recall.
I found that detail to be a massive conceptual hurdle at first, the idea that a gene dictates our personality.
It challenges how we view ourselves.
It really does. We are so conditioned to view our personality, our emotional state, as this ethereal thing.
Yeah.
It's our spirit, our soul, our lived experience, But this framework suggests our emotional regulation is deeply, inextricably tethered to a specific gene processing a specific chemical nutrient.
It is a profound realization. Your emotional state is not simply a reaction to the outside world. It is the direct result of physiochemical processes occurring in the brain. And this is where the data introduces a critical baseline for our discussion. Yes. MTHFR variation, or mutation, is one of the most common gene deficiencies on the planet. Millions upon millions of people possess a biological refinery that is operating with a broken catalytic cracker.
Wow.
They are taking in the raw crude oil, but they cannot efficiently convert it into the high-octane fuel.
Which introduces a massive contradiction in the clinical world, and I really want to examine the logic here. The analysis points out a deeply confusing clinical divide.
It's very polarizing.
Right. On one side, you have mountains of clinical evidence, randomized peer-reviewed trials demonstrating clearly that this genetic variation heavily impacts human health and cognitive function. But on the other side, there is a vocal contingent of clinicians who actively dismiss this gene variation.
They completely write it off.
They look at patients with an MTHFR mutation and tell them it's essentially harmless. How does a discrepancy like that exist in modern science? I mean, if this specific gene controls the active nutrients that literally build the neurotransmitters regulating our mood and focus, how could a medical professional ever categorize a deficiency at that foundational level as harmless?
If we look at the systemic structure of modern medicine, the mechanism behind that dismissal becomes much clearer. It really comes down to where a clinician is trained to look. Methylfolate is essential for DNA production, it's required for cellular repair, and crucially, it is the fundamental building block for neurotransmitter synthesis.
The chemical messengers of the brain.
Yes, but this raises an important question. Where are those chemical messengers actually synthesized? The vast majority of our neurotransmitters, the chemicals that drive our behavior, our processing speed, our emotional state, they are synthesized in the gut.
In the gut, not the brain.
Exactly. Modern medicine is often highly compartmentalized. If a patient presents with an inability to focus or crippling impulsivity, a psychiatrist is looking at the end-stage symptom in the brain. They are operating entirely downstream.
So they don't look at the gut at all?
Rarely. Their primary tool is to introduce a pharmaceutical stimulant to force the brain's remaining neurotransmitters to fire more aggressively. They are entirely focused on the behavioral output.
So they are basically treating the check engine light. They see the light flashing on the dashboard, so they put a piece of tape over it, or they rewire the bulb. They aren't opening the hood to realize the fuel line from the gut is fundamentally compromised.
That is the exact systemic blind spot. The MTHFR gene is the literal biological gatekeeper to mental well-being. It sits upstream of the entire cognitive process.
Right.
If a clinician is only trained to look at the brain, they will dismiss a genetic variation affecting gut-level nutrient synthesis as irrelevant. But the biology tells a different story.
Because if the gatekeeper is sluggish,
if the refinery is only operating at 30% capacity, the entire downstream system is starved of the chemical messengers it requires to maintain focus.
understanding that this gene is the upstream gatekeeper naturally forces us to ask an even deeper question about the mechanics. How precisely does this gene open or close the gates of our cognitive health on a microscopic level? Because the framework introduces a specific biochemical term for this process: methylation.
Yes, methylation.
And it sounds like standard textbook jargon, but understanding the actual geometry of it seems to unlock the entire ADHD puzzle.
Methylation is the absolute core of the biological engine. The data breaks it down to its most elemental microscopic essence. It's a biochemical process that scientists also refer to as one-carbon metabolism.
Okay, let's dissect one-carbon metabolism. What are we actually looking at under the microscope?
We are looking at one of the simplest molecular structures in organic chemistry. A methyl group consists of exactly one carbon atom bound to 3 hydrogen atoms. That is the entirety of it. CH3.
CH3. So it's tiny.
Incredibly small, and it is highly reactive. The process of methylation is simply the transfer of this tiny methyl group. It physically attaches itself to or detaches itself from another larger molecule, like a protein or a strand of DNA.
I was looking at the chemistry behind this, and it seems almost too simple to be the master controller of human biology. A cluster of 4 atoms jumps onto a protein. What does that physical attachment actually achieve structurally?
By attaching that tiny CH3 group, the shape and the electromagnetic charge of the larger molecule change slightly. And in biology, shape dictates function.
Right, the lock and key.
Exactly. When the shape changes, that methyl group acts as a master control switch. If it attaches to a specific segment of DNA, it can physically block the cellular machinery from reading that gene, effectively turning the gene off. It upregulates or downregulates a molecule's impact on the entire human system.
It's essentially a biological game of tag. Or, to visualize it another way, imagine looking down at the electrical grid of a massive metropolis. You have billions of microscopic light switches being flipped simultaneously every single second. A methyl group attaches a switch, flips on, an assembly line in the cell accelerates, a methyl group detaches a switch, flips off, and inflammatory response is shut down.
The light switch analogy perfectly captures the binary control mechanism. But to truly grasp the importance of this, we have to look at the sheer mind-bending scale of this biological electrical grid. The analysis provides a statistic that completely recontextualizes how we view our own bodies.
I know the number you're going to say, and it's crazy.
This specific process, this transferring of a carbon and 3 hydrogens, this flipping of microscopic switches occurs approximately 300 billion times a day in the human body.
300 billion times a day. It is a number so large it borders on the abstract. Every 24 hours, 300 billion distinct chemical transfers have to occur perfectly just to maintain homeostasis.
And those 300 billion actions are not optional. They are the fundamental engine of biological survival. We are talking about the continuous regulation of your gene expression. We are talking about deep cellular detoxification processes.
Like clearing out heavy metals.
Exactly. When a heavy metal enters your bloodstream, methylation is required to bind to it and excrete it. It regulates the metabolism of hormones. If this system falters, the biological grid begins to experience brownouts.
So let's draw the direct causal line from those 300 billion switches back to the person listening to this who struggles with ADHD. If I possess a variation in my MTHFR gene, a mutation that impairs my biological refinery, what is physically happening to my electrical grid?
The causal chain is deeply mechanical. If you have an MTHFR variation, your ability to convert raw folate into active methylfolate is severely reduced. Your biological refinery is inefficient.
Okay.
Because you lack methylfolate, your body lacks the actual methyl groups, the CH3 molecules, required to perform those 300 billion daily transfers.
So the physical inventory of light switches in my body drops. I simply do not have the molecular components necessary to flip the switches on.
Precisely. The overall efficiency of your one-carbon metabolism plummets, and the brain is highly sensitive to this drop in efficiency. The immediate consequence is that the synthesis of neurotransmitters in the gut begins to fail. The body cannot properly manufacture dopamine, and it cannot properly manufacture serotonin.
And right there is the holy grail of the ADHD conversation. Because dopamine and serotonin are not just abstract chemicals, they are the exact neurotransmitters that dictate the core battlegrounds of attention deficit challenges. Serotonin provides the biological sensation of satisfaction and calm. Dopamine is the molecule of pursuit, motivation, and sustained attention.
Exactly. When you lack dopamine, the brain feels starved of stimulation. It begins frantically searching for a hit of dopamine anywhere it can find it.
Which leads to the racing thoughts.
Leading directly to the racing thoughts, the inability to focus on a single task, and the profound physical impulsivity. It is a chemical starvation response.
This completely inverts how we try to solve the problem. If this microscopic engine is struggling because a genetic variation is choking off the supply of active methyl groups, the most logical first intervention is not necessarily to flood the brain with a synthetic pharmaceutical stimulant, to squeeze the last remaining drops of dopamine out of the synapses. The logical intervention is to change the raw fuel we are providing the refinery.
We have to look at the chemistry of the diet.
Exactly.
Which leads us to the most critical and perhaps most heavily misunderstood nutritional distinction in the entire analysis. We have to examine the profound structural difference between what we believe we are feeding our genetic engine and what we're actually putting into the tank.
The great folate divide. This is the exact moment in the framework where the biological mechanics translate directly into our daily lives. In common parlance, the terms folate and folic acid are used entirely interchangeably. Medical professionals, nutritionists, supplement labels, they use them as if they are the exact same chemical compound, but the data highlights a shocking fundamental distinction between the two.
The distinction is structural, and the biological consequences are massive. Folate is a natural, highly prevalent, essential nutrient. It is synthesized by plants. It has a specific three-dimensional geometry that the human body has evolved over millions of years to recognize and process.
Right.
Folic acid, however, is a completely different story. The framework states unequivocally that folic acid is a synthetic form of the vitamin that does not exist anywhere in nature.
It does not exist natively on the surface of the earth.
Yeah.
That is a massive red flag for biological compatibility. We have taken a naturally occurring molecular structure, altered it in a laboratory, and created a synthetic mimic. And our biological refinery, the MTHFR enzyme, which might already be struggling due to a genetic mutation, is suddenly tasked with processing a synthetic compound it has no evolutionary history of encountering.
The friction occurs at the enzymatic level. Enzymes are highly specific. They operate on a lock-and-key mechanism. The natural folate is the correct key. It fits perfectly into the MTHFR enzyme, the lock turns, and the conversion happens.
And the synthetic folic acid.
Folic acid is the synthetic key. It fits into the lock, but the geometry is slightly off. The lock refuses to turn easily. This forces the body to rely on secondary, highly inefficient enzymatic pathways in the liver to try and break down the synthetic folic acid. For someone with an MTHFR variation, this synthetic compound acts like a wrench thrown into an already slow set of gears.
And the true danger here is the sheer volume of exposure. We aren't just encountering this synthetic compound in a cheap daily multivitamin. The framework points a massive spotlight on the danger of what the food industry labels as enriched foods.
The terminology is incredibly insidious. When you walk down the aisles of a modern grocery store, the words fortified or enriched printed on a box of cereal sound like a premium health benefit.
Oh, absolutely. They imply that the manufacturer has gone out of their way to optimize the nutritional profile of the food for your benefit. Like, now enriched with essential B vitamins. It sounds like you're making the responsible, healthy choice for your family.
But the reality of that label is entirely different.
It's a marketing trick.
The reality is that the label is a mask for mass chemical intervention. The analysis explains that enriched, or fortified, means that highly processed, nutrient-stripped items like commercial white flour, white rice, white bread, white pasta, and breakfast cereals have literally been sprayed with synthetic folic acid.
So they strip the grain of its natural nutrient profile to increase shelf life, and they literally spray it with a synthetic chemical compound created in a lab. Let's combine these variables. If you have that anti-HFR genetic variation, your internal biological refinery is already operating at a deficit when trying to convert natural folate.
Right.
If you then consume a diet heavily reliant on enriched grains, you are dumping massive, unmanageable amounts of synthetic folic acid into the system. It must create a severe biological bottleneck.
It creates a phenomenon known in biochemistry as competitive inhibition. The massive influx of synthetic folic acid floods the bloodstream. These synthetic molecules bind to the cellular folate receptors. Because they are occupying the receptor sites, any actual natural folate you manage to eat cannot get into the cell.
Oh, wow.
The synthetic folic acid essentially barricades the door. It cannot be efficiently metabolized into methylfolate by the mutated MTHFR gene, and it prevents the good folate from doing its job.
That's devastating.
The methylation engine grinds to a near halt. Dopamine and serotonin levels crash. The ADHD symptoms, the brain fog, the emotional dysregulation, they all persist or significantly worsen.
Which brings us to the most actionable, empowering directive in this entire deep dive. The framework doesn't just outline the biochemical disaster and leave us stranded. It proposes a direct, highly practical intervention, a 7-day challenge. I want to speak directly to the listener for a moment, because this is where the theory becomes your reality. If you or if your children are actively battling ADD or ADHD, the framework recommends a simple, entirely harmless biological test.
And it's so simple to do.
Remove absolutely all fortified or enriched foods from your diet for exactly 7 days. Read every label. If it says enriched white flour, if it is sprayed with synthetic vitamins, throw it out for 1 week.
Yeah.
And meticulously observe the behavioral changes.
It is a brilliant exercise in self-quantification. The logic is biologically sound. If synthetic folic acid is acting as a competitive inhibitor, jamming the receptor sites and halting the production of neurotransmitters, removing that synthetic variable should yield immediate results.
Because it lets the system clear out.
Exactly. It allows the liver and the cellular machinery to clear the backlog of unprocessed synthetic compounds. You are lifting the chemical barricade, allowing the baseline genetic system to breathe and attempt to re-regulate its one-carbon metabolism.
But if we completely eliminate the synthetic mimics, we have a responsibility to flood the system with the authentic raw materials. We have to optimize our dietary fuel. What exact natural structure should we be introducing to support this delicate methylation process?
The dietary focus must shift entirely to whole foods that are naturally rich in bioavailable B vitamins, because B vitamins are the non-negotiable cofactors for the entire methylation cycle. We're looking at dark, leafy greens, spinach, kale, Swiss chard. We need complex legumes, lentils, chickpeas, black beans, and specific nuts and seeds, particularly sunflower seeds, which are dense with natural folate that the MTHFR enzyme evolved to recognize.
Real structurally intact foods carrying the exact molecular geometry the body expects. But the analysis includes a deeply specific detail regarding how we physically prepare these foods, particularly the leafy greens. And I love this part because it proves that optimizing biology isn't just about what you eat. It's about the physics and chemistry of how you process it before it enters your mouth.
Yeah.
You cannot simply graze on massive bowls of raw spinach and expect flawless absorption.
The thermal preparation is paramount. The text explicitly advises that spinach should be steamed before consumption to maximize its benefit for the methylation cycle.
I want to dig into the exact chemistry of that. Why steaming? What is the application of thermal energy actually doing to the cellular structure of the spinach leaf?
It all revolves around a specific organic acid present in the plant called oxalic acid, or oxalates. When a plant grows, it produces oxalates as a natural defense mechanism against pests and to manage its own calcium stores. Raw spinach is exceptionally high in oxalates. At a molecular level, oxalates are chelators.
Chelators, meaning they bind to things.
They possess a chemical structure that loves to bind with positively charged minerals, specifically calcium, magnesium, iron, and the crucial B vitamins we are trying to absorb.
They act like microscopic molecular handcuffs.
Precisely. When you eat raw spinach, the oxalates bind to the natural folate and the essential minerals, forming an insoluble crystalline complex. The human digestive tract does not produce the necessary enzymes to efficiently break down these oxalate crystals.
Wait, so what happens to the folate?
The folate remains locked in the handcuffs, and it passes entirely through your digestive system without ever entering your bloodstream. You ate the right fuel, but your biological engine couldn't access it.
It's the ultimate illusion of healthy eating. You think you're doing great with a huge raw spinach salad. But when we apply steam, when we introduce controlled thermal energy to the spinach, what happens to those handcuffs?
The heat fundamentally alters the cellular matrix of the plant. The thermal energy degrades the structure holding the oxalates in place. And because oxalates are highly water-soluble, the steam essentially leaches the oxalic acid out of the leaf and breaks those chelating bonds.
So it neutralizes it.
By lightly steaming the spinach, you neutralize the biological barrier. The natural folate is freed from the molecular handcuffs, rendering it highly bioavailable. When it reaches your small intestine, it is effortlessly absorbed into the bloodstream and delivered straight to the MTHFR refinery.
We are using heat to unlock the vault before we ingest it. That is a perfect example of applied biochemistry. So we've cleared out the synthetic folic acid, we are introducing the natural folate, and we are using thermal preparation to ensure total bioavailability. We are feeding the engine. But the brain is not just an abstract cloud of neurotransmitters. It is a dense physical organ.
Very much so.
It requires massive structural support, and it requires a cellular environment completely devoid of systemic inflammation to function, which means we must address structural fuel. Let's look at the sponsor product data from the source, analyzing it purely as factual biological interventions.
The physical architecture of the brain is largely composed of lipids or fats, and the framework highlights that one of the most structurally critical components for maintaining the integrity of the brain's cellular membranes is omega-3 fatty acids.
All right, this is the classic nutritional trope. Eat your fish. Salmon, mackerel, sardines. Everyone from cardiologists to psychiatrists pushes fish as the ultimate brain food. But the analysis throws a massive and frankly disturbing variable into this standard advice. It argues that simply eating fish is a biologically reckless game.
It really is.
The entire equation hinges completely on the specific environment where that fish was raised. Here's where it gets really interesting.
It is the stark division between wild-caught marine life and farm-raised aquaculture, and the biological implications of choosing the latter are profound.
The text is incredibly blunt about the grim reality of farm-raised fish. When you look at the mechanics of modern aquaculture, these fish are confined in incredibly dense, tightly packed underwater enclosures. Because of the sheer volume of biological mass in a stagnant area, the ecosystem collapses.
It's unavoidable.
The fish end up trapped in an environment where they're forced to constantly recirculate and ingest their own feces.
It is a scenario of extreme biological toxicity. You are taking an animal that evolved to swim across vast, pristine oceanic currents and confining it in a stagnant pool of concentrated biological waste.
And the framework specifically emphasizes the chemical fallout of that environment. That recirculated waste contains exceptionally high concentrations of toxic heavy metals. The fish are continuously breathing in and consuming these metals. Because heavy metals are lipophilic, meaning they bind to fat, these toxins become deeply and permanently embedded in the lipid tissue, the meat, of the farm-raised fish.
And that is the exact meat that ends up on the grocery store shelf.
Right. A consumer buys a massive fillet of farm-raised salmon, assuming they are investing in their cognitive health by securing omega-3s, but in reality, they are directly ingesting a concentrated dose of toxic heavy metals embedded within that fat. What happens to the human brain when a massive influx of heavy metals hits the bloodstream?
The body instantly recognizes the heavy metals as a severe threat to cellular integrity, and the immune system responds by triggering a massive systemic inflammatory cascade. Inflammation is the body's emergency alarm system.
Which normally is good, but in the brain.
But chronic inflammation in the brain, neuroinflammation, is completely catastrophic for cognitive function. The inflammatory cytokines physically disrupt the synapses. They alter the way dopamine and serotonin bind to receptors.
Even if your MTHFR gene is working.
Even if your MTHFR gene is producing enough neurotransmitters, a brain engulfed in neuroinflammation cannot utilize them. The environment becomes hostile to focus and conducive to severe impulsivity.
So the directive from the framework is an absolute strict requirement. If you are attempting to structurally rebuild the brain with omega-3s, you must secure wild-caught or line-caught fish. You must guarantee the structural lipids are free of heavy metals to prevent that inflammatory cascade at all costs.
And controlling that internal microscopic environment is a major theme of the analysis. The framework actually delves into two highly specific product interventions designed to aggressively optimize the cellular state. As a biological model, these are fascinating mechanisms. The first intervention targets the most fundamental aspect of our biology: hydration.
Hydration. Specifically, the introduction of high part per million hydrogen water via a system called the EcoGo Plus.
Right.
I want to really dissect the scientific premise here, because on the surface, hydrogen water sounds like a marketing buzzword. Water is H2O. It inherently contains hydrogen. What is the actual structural argument for infusing extra free hydrogen gas into the water we drink?
The clinical argument rests on the specific behavior of molecular hydrogen, or H2, within the human body. Because H2 is the smallest molecule in the universe, it has a unique biological property. It can effortlessly penetrate cellular membranes, and crucially, it can cross the blood-brain barrier with zero resistance.
It can slip right into the secure vault of the brain?
Exactly. And once inside the cells, molecular hydrogen acts as a highly selective antioxidant. It specifically targets and neutralizes the most toxic, damaging free radicals, like the hydroxyl radical, without interfering with the beneficial signaling molecules.
That's incredible.
By neutralizing these radicals, it radically reduces systemic and neurological inflammation. But the framework takes it a step further. It claims that this specific influx of hydrogen alters the redox state of the stomach, effectively balancing stomach acid and creating a vastly superior environment for nutrient absorption.
Absorption of the exact B vitamins and optimized omegas we've been curating. But the detail in the text that truly bridges this back to ADHD is the impact on the microbiome. The analysis asserts that this high part per million hydrogen water specifically feeds and optimizes an entire class of bacteria residing in the gut.
This is where the biological loop closes. Hydrogen acts as an electron donor for specific strains of beneficial gut microbiota. It provides the energy these bacteria need to flourish.
And as we established at the very beginning of this deep dive, the gut microbiota are the primary factory floor for neurotransmitter synthesis.
Precisely. If you are drinking water that actively acts as an electron donor for the exact bacteria responsible for manufacturing dopamine and serotonin, you are fundamentally reinforcing the foundation of your cognitive health. You are cultivating the biological soil.
Wow.
When the MTHFR gene provides the active methylfolate, the gut microbiome is primed, energized, and ready to produce the chemical messengers required to eliminate the brain fog and anchor your attention.
The elegance of that biological synergy is incredible. Optimize the cellular hydration, energize the gut bacteria, maximize the neurotransmitter yield. The second product intervention detailed in the framework focuses on the other vital raw material: amino acids.
Yes, the building blocks.
It discusses a full-spectrum essential amino acid profile called perfect amino. And I have to be completely transparent, when I was reading this section of the analysis, I experienced intense skepticism. It triggered a real moment of pushback for me.
Skepticism is essential when analyzing nutritional claims. What specific metric gave you pause?
The framework makes a staggering claim regarding standard protein supplements. It states that standard whey protein, which has been the undisputed, scientifically backed gold standard of the billion-dollar fitness and recovery industry for half a century, can be as little as 20% absorbable by the human body. I found that incredibly difficult to accept.
It's a huge claim.
We have built an entire global paradigm on the premise that chugging whey protein post-workout is the ultimate way to rebuild tissue. Does this analysis actually suggest that decades of standard fitness advice are biologically wrong, and that whey protein is fundamentally inefficient for cellular repair? How can it possibly justify a 20% absorption rate without completely defying established sports science?
The friction you are feeling comes from the difference between what standard sports science measures and what this framework is measuring. Traditional sports science often measures gross nitrogen retention to evaluate a protein. But this analysis is looking strictly at the enzymatic reality of biological utilization versus mere gastric intake.
Walk me through that.
We have to look at the structural complexity of whey protein. Whey is a massive, complex, folded, quaternary protein structure. When you drink a whey shake, your body cannot simply absorb that massive molecule into the bloodstream.
Right, it has to dismantle the structure first.
And that dismantling process is biologically exhausting. The stomach must secrete massive amounts of hydrochloric acid to denature or unfold the protein. Then, an enzyme called pepsin has to physically cleave the long protein chains into smaller peptides.
Sounds like a lot of work.
It is. Then, the pancreas has to release proteases to break those peptides down into individual elemental amino acids. Only then can the small intestine attempt to absorb them. During this massive, multi-stage, energy-intensive metabolic marathon, a massive percentage of the potential nutrient is lost.
Wait, really? Lost how?
If the gut is even slightly inflamed, which is common in modern diets, the enzymatic cleavage fails, and the undigested protein is converted into nitrogenous waste, which the kidneys must then filter out. That is the origin of the 20% utilization claim. The body burns so much energy trying to extract the aminos, the net cellular gain is minimal.
So it's like you are dumping a massive load of raw lumber onto the construction site, but you're forcing the carpenters to spend 80% of their energy just cutting the wood to size, leaving them exhausted before they even begin building the house.
A perfect analogy. And the framework contrasts that highly inefficient process with the mechanism of a full-spectrum, free-form amino acid product like Perfect Amino. The claim of 99% absorbability rests on the fact that these amino acids are already completely unbound. They are in their fundamental elemental form.
The lumber is already cut to the exact millimeter required by the cellular blueprints.
Exactly. There is zero enzymatic cleavage required. The stomach acid doesn't have to unfold anything. The pancreas doesn't have to release proteases. The free aminos pass directly through the mucosal lining of the small intestine and straight into the bloodstream within minutes.
And because there is no massive metabolic breakdown process, the analysis notes that it has virtually zero caloric impact on the body. It operates at something like 2 calories per dose, meaning it can facilitate massive cellular repair without even breaking a metabolic fast.
Which is incredibly efficient.
But how does this deep dive into muscle recovery translate to the ADHD brain? Why is an ultra-absorbable amino acid profile non-negotiable for someone with an MTHFR variation?
We have to zoom back out and look at the fundamental equation of the MTHFR gene. Its entire job is to take an amino acid and convert folate into methylfolate. Furthermore, what are neurotransmitters made of?
Amino acids.
Dopamine is synthesized from the amino acid tyrosine. Serotonin is synthesized from the amino acid tryptophan. Amino acids are the literal physical building blocks of the chemical messengers. If your body is burning all its energy trying to cleave massive whey proteins and your inflamed gut is failing to absorb the specific aminos you need, your biological refinery is starved of its structural components.
So by introducing a 99% absorbable, free-form amino acid matrix, you're instantly flooding the bloodstream with the exact molecular raw materials the MTHFR engine needs to execute the methylation cycle.
And build the neurotransmitters that stabilize focus.
Okay, let's step back and look at the massive biological fortress we have just constructed. We have engineered the optimal fuel line. We have identified and eliminated the synthetic folic acid that causes competitive inhibition. We are utilizing thermal energy to break the oxalate bonds in our leafy greens, flooding the system with bioavailable natural folate.
Right.
We are sourcing wild-caught marine lipids to prevent heavy metal neuroinflammation. We are saturating our cells with hydrogen electron donors to energize the gut microbiome, and we are bypassing the enzymatic bottleneck with highly targeted, free-form amino acids. The system is fundamentally optimized.
The architecture is sound. However, a fortress is ultimately only as impenetrable as its defenses against infiltration.
And that reality forces us into the next critical phase of this analysis, because we exist in an environment saturated with hidden saboteurs. There are dietary inclusions in our daily lives, some completely ubiquitous, and others universally praised as healthy, that possess the chemical ability to bypass our biological fortress and completely derail the delicate genetic balance we just painstakingly built.
The framework pivots to address the severity of modern processed food, but it bypasses the standard, tired arguments about empty calories or saturated fats. It targets a very specific, heavily engineered additive, artificial colors.
This was another moment in the text that forced me to stop and reread the paragraph. I think the vast majority of consumers view artificial food dyes, red dye 40, yellow 5, blue 1, as completely inert, harmless visual aesthetics.
Just pretty colors for kids?
Yeah. We assume they are just chemical pigments added to cereal or sports drinks to make them visually stimulating, but the analysis unveils a terrifying biological reality. It states that artificial colors are scientifically classified and proven to act as antimicrobials.
We must seriously pause and analyze the functional definition of an antimicrobial. The literal purpose of an antimicrobial agent is to actively seek out, disable, and kill microscopic life. It is designed to eradicate bacteria.
Which is fantastic if you were designing a chemical to sterilize a stainless steel surgical table. But it is an absolute biological catastrophe if you are voluntarily ingesting that chemical into your highly delicate intestinal tract.
It is chemical warfare against your own biology. To understand the severity, we have to look at the mechanism. How does a dye kill a bacteria? Many of these synthetic dyes are built with complex aromatic rings and petroleum-derived molecular structures. When these structures enter the gut, they physically integrate into the lipid bilayer of the bacterial cell membranes. They alter the cell's permeability, disrupting its metabolic engines until the bacterial cell essentially ruptures and dies.
And we just spent the last 20 minutes meticulously explaining how the gut microbiota is the absolute epicenter of neurotransmitter production. The bacteria in our gut are the factory workers synthesizing the dopamine and serotonin we need to manage ADHD.
Exactly.
If you consume a sports drink loaded with artificial colors, you are effectively dropping an antimicrobial bomb directly onto your own biological factory floor. You are chemically assassinating the exact bacteria required to maintain your cognitive focus.
The destruction of the microbiome halts the synthesis of neurotransmitters instantly. This is why the analysis states that artificial colors can exponentially exacerbate MTHFR symptoms. The genetic mutation is already struggling to fuel the process, and the artificial dye burns the factory to the ground. It is an insidious, multi-front attack on cognitive stability.
And right alongside the synthetic dyes, the framework isolates the classic, undeniable saboteurs: high refined sugar and alcohol. But again, it doesn't just offer the generic sugar is bad platitude. It provides the exact mechanical pathways demonstrating why these specific molecules are catastrophic for this specific genetic profile.
The danger of refined sugar lies in its disruption of the endocrine system, specifically insulin regulation, and how that cascades into the methylation cycle. When you consume a massive load of refined sugar, you flood the bloodstream with glucose,
Which triggers insulin.
the pancreas must aggressively spike insulin levels to force that glucose into the cells. This creates a massive, volatile spike in cellular energy, inevitably followed by a profound, rapid crash as the glucose is cleared.
The classic sugar crash. But how does that blood sugar roller coaster interact with the MTHFR gene?
The methylation cycle is a highly sensitive, continuous biological engine. It requires a stable, unbroken supply of energy ATP to perform those 300 billion daily chemical transfers. When your blood sugar is violently spiking and crashing, the cellular energy required for methylation becomes highly erratic.
That makes sense.
Furthermore, massive insulin spikes trigger low-grade inflammatory cascades, which, as we established, disrupt the brain's ability to utilize whatever dopamine the sluggish MTHFR gene manages to produce. It completely destabilizes the delicate environment the genetic engine requires to function smoothly.
And then we must examine the chemistry of alcohol. The analysis makes a very specific point that individuals harboring the MTHFR gene variation are disproportionately and severely affected by alcohol consumption. What is the molecular mechanism driving that heightened sensitivity?
The mechanism lies entirely in the byproduct of hepatic or liver metabolism. When you ingest ethanol, the active alcohol in beverages, the liver uses an enzyme called alcohol dehydrogenase to break the ethanol down. The immediate resulting chemical compound is called acetaldehyde.
Acetaldehyde.
Acetaldehyde is a highly reactive, deeply toxic molecule. It loves to bind to cellular proteins and DNA, causing structural damage. But critically, the analysis points out that a buildup of acetaldehyde physically alters the acid-base balance of the body. It makes the blood plasma slightly more acidic.
Wait, it shifts the entire pH of the human bloodstream.
Yes. The human body is designed to maintain a strictly controlled blood pH between 7.35 and 7.45. Even a microscopic shift toward acidity causes a systemic panic. Enzymes are highly sensitive to pH levels. If the blood and the brain's cellular environment become even slightly acidic due to acetaldehyde, the shape of the enzymes begins to warp.
And its shape dictates function.
The MTHFR enzyme, which is already structurally compromised by the genetic mutation, loses even more of its binding efficiency in an acidic environment.
The broken lock becomes even harder to turn.
Precisely. Furthermore, the body relies heavily on the methylation cycle to detoxify and excrete that toxic acetaldehyde. But if your MTHFR gene is mutated, your methylation cycle is slow. So the toxic acidic byproduct lingers in the system far longer than it should, causing prolonged neuroinflammation, severe cognitive fog, and a massive amplification of the impulsivity and inattention characteristic of ADD and ADHD.
So the biological math is undeniable. Eliminate the sugar and aggressively moderate or entirely abandon the alcohol to protect the enzymatic environment. But that leads us to the most beloved chemical dependency in modern society.
Coffee.
Coffee. If we are advising a listener who relies on heavy stimulation to manage their ADHD that they must suddenly abandon their morning coffee, the entire framework might be rejected on principle.
The framework provides a very welcome, chemically sound caveat here. The stimulant molecule itself, caffeine, is not the enemy. The text notes that an average cup of coffee contains between 85 and 200 milligrams of caffeine. And when we look at the clinical data, there is no indication that sustained long-term intake of caffeine at that specific dosage has detrimental effects on the methylation cycle or the MTHFR gene.
That is a massive relief. But the framework doesn't offer blanket immunity to coffee, does it? There is a massive structural catch regarding the sourcing of the bean.
The danger is not the caffeine, the danger is the agricultural reality of the coffee crop itself. The analysis warns that non-organic, mass-produced coffee carries an exceptionally high risk of mycotoxin, or mold, toxicity.
Mold in the coffee.
Yes, coffee plants are heavily sprayed with pesticides, which stresses the plant. More importantly, during mass harvesting and bulk storage in damp environments, the beans frequently develop microscopic molds. These molds release mycotoxins into the bean. When you brew the coffee, you extract the mycotoxins and ingest them.
And mycotoxins, much like heavy metals, are an immediate trigger for a massive immune response. The body senses the fungal toxin, the inflammatory cascade initiates, and the brain is bathed in neuroinflammation, entirely negating any cognitive benefit the caffeine might have provided.
Completely negated.
So the directive is clear. If you rely on the dopaminergic stimulation of coffee, you must invest the resources to procure certified, organic, rigorously tested, mold-free beans. But the detail in this section that genuinely shocked me was the analysis of decaffeinated coffee. Logic would dictate that removing the stimulant makes it the safer, more biologically gentle choice. But the framework completely dismantles that assumption by exposing the industrial processing required to remove the caffeine.
It's a harsh chemical process.
It highlights that many commercial decaffeination processes utilize literal formaldehyde to strip the caffeine from the bean.
Formaldehyde, a highly volatile, toxic organic compound, primarily utilized as an industrial embalming fluid and tissue preservative.
It is a stunning realization of how blind we are to industrial food chemistry. A consumer trying to manage their anxiety or racing thoughts makes the conscious choice to cut out caffeine. They switch to decaf, assuming they are acting in the best interest of their nervous system. Instead, they are microdosing their biological engine with a highly carcinogenic, profoundly toxic chemical solvent.
It's tragic, really.
And if that individual possesses an MTHFR variation, their primary detoxification pathways, their methylation cycles, are already critically impaired. You are introducing a severe cellular toxin into a body that lacks the chemical machinery to excrete it. It is the absolute worst-case scenario for neuropreservation.
It perfectly underscores the core philosophy of the analysis. You cannot just look at the base ingredient. You must deeply understand the chemical and industrial journey that ingredient took before it entered your biology. And this principle brings us to what I consider the most fascinating, counterintuitive, and intellectually satisfying revelation in the entire framework.
The green tea paradox. Throughout this entire discussion, we have been identifying obvious chemical villains. Synthetic folic acid, farm-raised feces, artificial dyes acting as antimicrobials, acidic alcohol, embalming fluid in coffee. These are all intuitively destructive. But green tea...
It's the ultimate health drink.
Green tea sits at the absolute pinnacle of universally celebrated human health. Every dietitian, every longevity expert, every ancient medical tradition hails green tea as a pristine elixir packed with life-extending antioxidants.
What's fascinating here is that universal praise is exactly what makes the genetic reality so interesting, because the framework introduces a secondary genetic variable into the equation. Up until this point, we have focused exclusively on the MTHFR engine, but the human genome is a vast interconnected network. The analysis brings our attention to a different gene known as the COMT gene. COMT.
Let's define the parameters of the COMT gene. What is its specific role in the neurochemical factory?
COMT stands for catechol-methyltransferase. While MTHFR is responsible for helping to build neurotransmitters, the COMT gene produces an enzyme whose primary biological directive is to degrade and clear out specific neurotransmitters known as catecholamines once they have been used.
So it's the cellular cleanup crew.
Exactly. Its main targets are dopamine, epinephrine, and norepinephrine, essentially the excitatory, stimulating neurotransmitters.
So MTHFR helps build the dopamine to help you focus, and COMT sweeps the dopamine away when it's time to relax. What happens when a person has a mutation or a variation in this specific COMT gene?
If you possess a slow COMT mutation, your cleanup crew is highly inefficient. It takes much longer for your brain to clear out the excitatory neurotransmitters. Your baseline levels of dopamine and adrenaline remain elevated far longer than a typical biological profile.
And the framework reveals that if you possess this specific slow COMT mutation, the universally lauded advice to consume green tea is completely devastatingly inverted.
It becomes an active sabotage of the nervous system. The molecular reason lies in a specific, highly concentrated chemical compound found in green tea called quercetin. Quercetin is a powerful polyphenol. For a biological profile with standard genetics, quercetin acts as a brilliant antioxidant. However, quercetin has a highly specific interaction with the COMT enzyme. It is a potent COMT inhibitor. It physically binds to the COMT enzyme and stops it from working entirely.
Let's trace that cascade, because it is mind-blowing. You have a slow COMT gene, meaning your brain is already struggling to sweep away the adrenaline and dopamine. You drink a massive cup of green tea, flooding your system with quercetin.
Right.
The quercetin seeks out your already struggling COMT enzymes, binds to them, and shuts them down completely. The cleanup crew is essentially fired and sent home.
The result is a massive, unchecked biological backlog of excitatory neurotransmitters in the synapses. The adrenaline and dopamine have nowhere to go. They continue to violently fire against the receptors.
The framework describes the precise, lived experience of this chemical interaction, and it is eerily specific. It details the exact phenomenon where an individual lays down in bed to go to sleep. The physical environment is completely silent, the lights are off, the external stimuli are removed, but internally, the mind suddenly, violently wakes up.
The racing, inescapable waking mind at 2 in the morning. It is the hallmark symptom of neurodivergent ADHD-like struggles and severe anxiety.
It's like drinking a celebrated health elixir, only to find out your specific genetic blueprint interprets it as a shot of adrenaline right before bed. They are chemically inducing our racing mind because their mutated COMT gene is being suffocated by the quercetin.
It is the ultimate, undeniable proof that the one-size-fits-all approach to diet, nutrition, and cognitive health is biologically illiterate. What acts as medicine for one genetic profile acts as a neurochemical poison for another.
Okay, the mechanical elegance of this entire framework is stunning. The genetic switches, the methylation cycle, the competitive inhibition, the oxalate handcuffs, the COMT enzyme. It weaves an incredibly cohesive, logical biological narrative. But as we synthesize all this data, we must demand empirical validation. We have to ask the critical question: how deeply grounded is this narrative in actual, rigorous scientific literature?
Very grounded.
Is this just a highly convincing theoretical model of biohacking? Or is there unassailable clinical data proving this mechanism dictates ADHD?
The framework does not rely on theory. It rigorously anchors these biochemical claims in hard clinical evidence. The link between the MTHFR genetic mutation and the presentation of ADHD has been extensively mapped by the scientific community. The analysis specifically highlights a massive foundational study, the meta-analyses conducted by Go et al. In 2018.
We need to define the weight of a meta-analysis for the listener because it is the gold standard of scientific proof. A meta-analysis is not a single, isolated experiment with 50 college students in a controlled room.
No, a meta-analysis is the apex of evidence-based medicine. It is a comprehensive statistical aggregation of data pulled from dozens, sometimes hundreds, of independent, rigorous studies across the globe. It examines massive, highly diverse data sets to filter out anomalies and identify overarching, undeniable statistical realities.
And what were the results?
And the results of the 2018 Go et al. Meta-analysis were unequivocal. They found a mathematically clear, undeniable association between MTHFR polymorphisms, meaning the specific variations and mutations in the structure of the gene, and a significantly increased risk of developing the pathology we label as ADHD.
The statistical correlation is proven. The genetic link is not a theory, it is a mapped reality. But the framework pushes beyond statistical correlation. It introduces a highly specific biological marker that any clinician can actually measure. It provides a literal, measurable smoking gun floating in the bloodstream, a molecule called homocysteine.
Homocysteine is the final critical piece of the methylation puzzle. It is an amino acid naturally produced within the body as a byproduct of cellular metabolism. Under optimal, healthy genetic conditions, homocysteine is a transient molecule. The body uses the methylation cycle, the active methylfolate, produced by the MTHFR engine, to instantly bind to the homocysteine and recycle it back into a beneficial amino acid called methionine. The system is designed to clear it continuously.
But what happens when the 300 billion switches aren't flipping? What happens when the MTHFR gene is mutated, the active methylfolate is absent, and that specific chemical recycling pathway is broken?
Because the body lacks the methyl groups to convert it, it loses the ability to break the homocysteine down. The molecule becomes trapped in its current state. As cellular metabolism continues, more and more homocysteine is produced, and the levels in the blood plasma begin to rise dramatically.
The physical concentration of the molecule spikes. Why is elevated homocysteine a biological crisis?
Because unbound homocysteine is highly toxic to the human vascular system. As it accumulates, it physically damages the fragile endothelial lining of the blood vessels. It induces massive oxidative stress. The analysis directly links elevated homocysteine to dangerous hypertensive episodes, severe spikes in blood pressure.
And for the brain.
But crucially for the cognitive conversation, the clinical data proves that elevated homocysteine levels in the brain are directly, inextricably linked to a severe exacerbation of ADD and ADHD symptoms.
I want to extend our earlier refinery analogy because it perfectly encapsulates this danger. If the MTHFR gene is the crude oil refinery, homocysteine is the toxic, thick, black exhaust smoke produced by the engine. In a healthy system, the ventilation fans, the methylation cycle, blow the exhaust out of the factory instantly. But if the fans are broken due to the genetic mutation, the toxic exhaust begins to rapidly accumulate inside the enclosed factory floor. It eventually chokes the workers, halts the assembly line, and the entire cognitive production system suffocates. Homocysteine is the exhaust fumes of a broken genetic engine.
That is a flawlessly accurate biochemical visualization. The buildup of the exhaust directly impairs the brain's ability to utilize neurotransmitters.
So if elevated homocysteine is the scientifically measurable smoking gun of a broken engine, what is the clinical intervention? How do we chemically force the ventilation fans back on and clear the exhaust from the brain?
The clinical studies highlighted by the framework point to a highly specific, targeted molecular intervention. The solution is to artificially supply the specific molecular groups the body is failing to produce. The data shows that introducing targeted supplementation specifically, utilizing trimethylglycine, known as TMG, alongside pure active methylfolate provides a direct biochemical override.
Let's look at the chemistry of that intervention. Methylfolate, that is the exact, active, highly refined fuel we discussed at the very beginning of the deep dive. The fuel the broken MTHFR gene is struggling to make.
Exactly. You stop relying on the broken refinery to process raw folic acid. You bypass the malfunctioning enzymatic machinery entirely and deliver the final, highly refined, combustible methylfolate directly into the bloodstream. Furthermore, trimethylglycine, or TMG, is a molecule that literally carries 3 active methyl groups. It acts as a massive molecular donor.
You are handing the biological grid a massive crate of pre-assembled light switches.
Precisely. By flooding the system with TMG and methylfolate, you provide the exact chemical components required to restart the recycling process. The homocysteine is rapidly bound, converted, and cleared from the blood. The exhaust fumes are evacuated from the factory.
It's incredible.
And the clinical outcomes are stunning. As the homocysteine levels drop back to a baseline state, the clinical impact of the ADD and ADHD symptoms lessens dramatically. Focus returns, impulsivity drops, and the internal motor calms down.
The clarity of that biological sequence is staggering. Bypass the broken gene, deliver the active molecule, clear the toxic exhaust, and restore cognitive function. So as we pull back the lens and look at the entirety of what we've explored today, what does this ultimately mean for the individual listening to this?
It changes everything.
It really does. We have traversed a massive landscape of microscopic biology. We have dissected the enzymatic conversion of crude folate, the 300 billion daily transfers of carbon and hydrogen atoms, the competitive inhibition caused by synthetic sprayed-on vitamins, the necessity of thermal degradation for oxalates, the heavy metal neuroinflammation from farm-raised feces, the antimicrobial nature of artificial dyes, the pH disruption of acetaldehyde, ketoaldehyde, and decaf, and the paradoxical sabotage of green tea by the KOMT gene. It is a vast, complex molecular web.
But it all points to one reality.
Yes, if we distill all of this data into a single, cohesive reality, it means that managing focus, reclaiming your attention, and defending your cognitive health is absolutely not a matter of psychological willpower. It is not a test of discipline. It is the practice of actively, intelligently downregulating a hostile internal biological environment through the application of highly specific nutritional chemistry.
It demands a profound, foundational shift in our entire cultural perspective. We are actively moving away from the archaic view of ADHD as a fixed, permanent behavioral flaw.
Wow.
We are rejecting the narrative that a racing mind is just the inevitable, tragic consequence of trying to exist in a modern, hyper-digital reality.
Right.
We are now empowered to understand these cognitive struggles strictly through the lens of individual mechanical genetic variations like the MTHFR mutation and the critical, non-negotiable availability of specific chemical nutrients.
It completely shifts the locus of control back to you, the listener. It is a deeply empowering framework. because you now possess the biochemical literacy to look at the physical world around you with entirely new eyes.
You really do.
Tomorrow, you can walk down the aisle of a grocery store, pick up a brightly colored box of enriched cereal, and instead of seeing a harmless, convenient breakfast, you will see a payload of synthetic folic acid that will cause competitive inhibition and jam your genetic receptor sites. You can look at a slab of farm-raised salmon and visually trace the inflammatory cytokine response it will trigger in your synapses. You are no longer navigating blindly at the mercy of vague, overwhelming behavioral symptoms. You understand the microscopic chemistry driving them.
You have acquired the biological tools to make proactive, highly specific, targeted choices that align flawlessly with the reality of your unique genetic blueprint.
Which brings us full circle back to the very first image we discussed, that broken X-ray machine, the murky, confusing diagnostic waters of ADHD. Through the lens of this analysis, we've essentially discovered that the X-ray machine was never broken at all. The medical establishment was simply looking at the wrong kind of film. We spent decades searching for a behavioral fracture, when we should have been looking at the chemical reality of a genetic nutrient deficiency.
It fundamentally changes the entire diagnostic paradigm. And as we conclude this deep dive into the microscopic mechanics of the human brain, there is one final, highly provocative thought derived from this framework that I believe requires deep reflection.
Let's hear it.
We learned today, through the undeniable chemistry of the COMT gene, that a universally celebrated, historically lauded health practice, the daily consumption of green tea, can chemically trigger severe neurodivergent, anxiety-inducing symptoms in people harboring a specific genetic variation. It physically prevents the clearance of adrenaline, keeping the mind violently racing all night long.
A celebrated elixir of life acting as a direct neurochemical poison for a specific genetic type.
Exactly. So I leave you with this conceptual challenge to ponder as you navigate the modern wellness landscape. If a single variation in a single gene can completely structurally invert the definition of what constitutes a healthy food, how many other universal blanket dietary guidelines relentlessly pushed by the modern nutrition industry might be fundamentally, biologically incompatible with the unique genetic blueprint sitting inside your own cells?